and welcome to the latest Down the Rabbit Hole interview. Today we're going to be talking about the respiratory microbiome and what it means for our immune system. I'm joined today by Dr. Aran Singanayagam. Aran, could you please introduce yourself and tell everyone a little about what you do?
Yeah, so hello everyone, many thanks for inviting me. My name is Aran Singanayagam. I am a clinical academic working at Imperial College. So I'm a doctor by training, so part of my week is spent seeing patients in clinic, and then the other major component of my week involves running a research lab at Imperial College where we study the immune system and specifically how the airway microbiome impacts upon that. And we're particularly interested in chronic lung diseases, including asthma and COPD.
Fantastic, thank you so much. And so if we dive straight into the microbiome, it's more consciously entered the public sphere in recent years, but mostly in terms of the gut microbiome. Can you tell us a bit about the history of the discovery of the respiratory microbiome? How long have we known about it?
Yeah, so it's quite interesting because if you sort of read old medical textbooks, talking about the lungs and the lower airways, as we call it, the old textbooks sort of over 10, 15 years ago now would tell you that the lungs are completely sterile, so that they don't contain any bacteria whatsoever in health. Now, obviously, looking back on that now, we now know that not to be the case. We've applied more sensitive molecular techniques to studying the lungs, and we found that there's clearly a microbiome, communities of commensal bacteria that reside in the lungs, even in healthy people. But as I said, the dogma in textbooks of yesteryear was that the lungs are sterile. And I think when we look back with the benefit of hindsight, you would probably imagine that notion that the lungs are sterile was likely to be false, because even if you take soil samples from Antarctica, for example, so really, really harsh climates, much harsher than the climate of the lungs, you can still recover bacteria from those types of samples. So it was probably pretty implausible that the lungs were sterile, but certainly that was what everyone thought back in the day. But now, as I say, we've applied sensitive molecular techniques, and what's clear is that there's definitely a microbiome in the airways as there is in the gut.
Fascinating. So life finds a way. Evidently, it's a growing field. How much is our pre-existing understanding of the gut microbiome influencing and informing this as we grow?
Yeah, I think we're very lucky in the airway microbiome field that we have the gut microbiome field, which is several decades, ahead in our understanding. And I think there's clear similarities between the two. We know from gut microbiome studies that the microbiome is a major player in regulating immunity, and the gut microbiome field has got so far forward that we now have therapies where you can manipulate the gut microbiome to influence the cause of disease. With the lung microbiome, I think there are a few differences between the lung and the gut. One thing to sort of be aware of is the fact that the amount of bacteria that are present in the lungs is a lot lower than it is in the gut. So therefore, directly extrapolating what we know about the gut to the lungs is perhaps not going to be completely correct, but I think there's clear similarities and areas of overlap where the two microbiomes influence similar processes. So I think we are lucky that we've got the gut microbiome field to guide us, but at the same time, it is a brand new field, and we do need to understand specifically what the lung microbiome does.
So let's dive into that then. So I know that your work overlaps significantly with the immune system and immune responses. So what insights can you say about the microbiome changing or influencing our immune responses?
Yeah, so I think because we're quite early in our sort of understanding of the lung microbiome, in terms of sort of functionally what it does, we're a little bit sort of still in the dark. So most of the studies that have been published, and there have been lots of studies now, have really taken samples from human patients and often compared the microbiome of a disease with the microbiome of a healthy individual, so COPD versus health, asthma versus health, etc. And you see profound differences between diseased individuals versus healthy individuals. What we don't know is whether those changes are just a consequence of the diseases or if they play a direct causal role in people developing the disease or in driving the complications of the disease. And that's sort of one of the main areas that we're trying to understand within my group. We know from the gut microbiome field that the only way that you can really understand function is to try and manipulate the microbiome. So you deplete or change the commensals present and then look at what that does to disease processes or to the immune system and the immune response. So we're trying to mimic what has been done in the gut microbiome field within the lungs, and part of that obviously involves work in animal models, so particularly in mice, where we're looking to try and sort of alter the microbiome, the lung microbiome of mice, for example, using antibiotics delivered into the airways, and then looking at what that does to, for example, the immune response to a viral infection, because that's sort of one area that we're particularly interested in. So I think, sort of in answer to your question, I think we're sort of still pretty early in our understanding of functionally what the lung microbiome does. We can be guided a little bit by what the gut microbiome does because the types of commensals are very similar. But we really need that sort of body of functional experiments of the sort that I've described before we can really kind of understand what's going on with that.
That's fascinating. And do you see actually AI coming into that as well? You've talked about animal models. Can you see that being useful?
I think AI is useful in terms of sort of microbiome analysis. So when you get samples either from mice or from human patients, obviously they contain a lot of different types of bacteria, and sort of sifting through the huge amounts of data you get from sequencing studies, which is how we characterize the microbiome, using kind of artificial intelligence type approaches. It can be very useful because obviously you need that sort of bioinformatic power to be able to sift through the data and find correlations or find important commensal bacteria that associate with disease, and that's not easy to do without a sort of powerful type approach. So I think AI comes into play there within the sort of data analysis side of things.
That's fascinating. I'm seeing a real huge push for AI or help that AI is supporting in genomics overall. So great to know it's helping here as well. And what are our current approaches and techniques for treating common respiratory disorders? And what are the problems with the current approaches? How can an increased understanding help?
I think within respiratory medicine, we're really sort of way behind other specialities in terms of being able to treat our patients. I'll give you an example of COPD, chronic obstructive pulmonary disease. That's sort of one of the conditions that I study a lot. It's obviously a smoking related condition, and we don't have very many treatments at all. We've been using the same treatments, that's mainly inhalers, for the last several decades. We haven't had any new effective licensed therapies for COPD. We don't have any therapy that changes the course of disease. We don't have any therapies that can restore damaged lung, because obviously these people have very damaged lungs. We also don't have effective therapies to prevent infections. Infection is a major problem in COPD. These people are prone to developing viral infections, bacterial infections, and we don't really have an effective way of stopping them. So I think our current approaches are extremely limited, and as I say, sort of mainly focused around giving inhalers, which have pretty modest benefits.
Okay, so how can an increased understanding of the microbiome, how might that change our approach?
Yeah, so we believe that targeting the microbiome could be a really effective means of trying to address some of those unmet needs that I've described. As I said earlier, we know that the airways of patients with COPD have altered microbiota, perturbation of the microbiota compared to health. And I also said that, you know, that could either be a consequence or a cause of the disease. And obviously, if it's just a consequence of the disease, then targeting that would not be that, you know, may not be that useful. What we're finding with some of the work we're doing, both in animal models and in human samples, is that we think the microbiome is a major player in disease pathogenesis. So it's a sort of causal driver. As an example, there's one particular type of bacteria called proteobacteria that are very commonly expanded within the airways of COPD patients, and we know that patients that have you know, huge expansion of proteobacteria are often very prone to developing viral infections and what we call exacerbations or flare-ups. So it's a very strong risk factor for being susceptible to viruses. So that's pretty good evidence that perhaps the composition of the microbiome could be important in driving the course of disease. We're also seeing in our animal models, as I say, where we're trying to manipulate the airway microbiome, that when we manipulate the mouse airway microbiome to mimic what we see in patients, that we see that similar susceptibility to severe viral infections when we then challenge those mice with viruses. So I think we're getting, with some of the work we're doing currently, we're getting some pretty clear signals that the microbiome is a major determinant of of all the complications of COPD and other chronic lung diseases, and therefore it's a viable therapeutic target for the future. I think the other thing to point out is that these changes, for example, the expansion of proteobacteria, that's seen in all lung diseases. So it's not just in COPD, it's seen in asthma, it's seen in lung fibrosis, it's seen in cystic fibrosis. So you've got a real, potentially targeting that abnormality, you've got a real opportunity to change the treatment of multiple chronic lung conditions.
Wow, a huge potential impact there then.
Yeah.
And I know you've just talked about bacterial and viral infections. Are fungal infections forming part of the picture?
Yeah, so the other thing that I haven't already mentioned is that as well as there being commensal bacteria, which, traditionally when we talk about the microbiome, we're talking about bacteria. But there's also other commensals within the lungs as well, including viruses, so the virome as we call it, but also fungal commensals, the microbiome as we call it, or the fungome as some people might call it. Now we understand even less about those commensal bacteria than we do about bacterial commensals, but we think also that they're likely to be highly important as well. And we've got a stream of work going at the moment where we're characterizing and studying those fungal commensals within the lungs, seeing how they change in chronic lung diseases such as asthma and COPD, and then similarly seeing how they affect immune responses to other pathogens, including viruses. So that's sort of commensal fungal organisms. You then obviously have pathogenic fungi, which can also be a trigger of flare-ups of the disease as well. So again, that's a sort of another layer to explore as well.
Fascinating. And what are the challenges of collecting samples?
So the airways is a challenging site to collect samples from, more so than the gut. Obviously, in the gut you can collect people's poo, and it's easy enough, and that's quite a deep sample that you're getting. The problem with the airways is we sort of have two ways of doing it. So you can either take what we call a sort of non-invasive sample, so that would be typically sputum. For patients that can cough up sputum, they can give us a sputum sample. That's not invasive. They just cough it into a pot and they give it to us. And the other major sort of way of looking at the microbiome in the less invasive or a non-invasive way would be throat swabs, so a swab at the back of a patient's throat, or also we can look at the nasal microbiome. So obviously the nose is a continuum of the lungs, so you can study the nasal microbiome very easily by washing the nose out and taking sort of what we call lavage samples. Now the problem with those types of samples is that you get a lot of contamination because the upper airway, excuse me. So the upper airway is in close contact with the oropharynx, where food goes down. So if someone coughs up a sputum sample, for example, it's going to go up through the lungs, but it's also going to go through the throat as well and pick up contaminants. So you need to be careful about your analysis. You need to look for these types of contaminants. And sometimes it's difficult to know what is a contaminant, because obviously a lot of what's in the lung microbiome comes from the oropharynx. So it's sort of what we call aspirate bacteria from the gastrointestinal tract into the lung. So there's a lot of overlap anyway. So you don't know what's real and what's contaminant. So that really the best sample type is a far more invasive sample, where we do a test called bronchoscopy, where we pass a telescope down through the nose into the lungs, and we can then take deep targeted samples, and we can take washings, we can take small pieces of tissue, and we can study the microbiome in those types of samples. Those samples are far more representative of what's going on in the lungs, but as you can imagine, that's obviously quite an invasive test. You have to bring the patient into hospital, you have to give them a sedative, you have to, it's not a very pleasant test to have. There are risks associated. It's an invasive procedure. So it's not easy to get those types of samples. We do get them, but it's much easier to look at sort of sputum and nasal samples, as I mentioned. But you then do have those caveats that I mentioned. So I think getting airway samples is in general quite tricky, and sometimes you have to be pragmatic and use perhaps not the optimum sample, but the sample that you can easily get from the patient.
Absolutely. And another lens on the sample question, I know that in genomics, we often end up with a diversity problem in regards to the populations we're sampling and accessing. Obviously, this is a very new field. Where are we currently, and where do you see us going in regards to the diversity of our samples as well?
Do you mean sort of inter-individual diversity? So you take a sample from one person and there's so many different confounding factors versus another person? Is that what you mean? Or?
Yeah, so demographic diversity, international diversity.
Yeah, absolutely. So yeah, that's a thing that we don't really understand fully. I mean, you take a sample from patient X that lives in the country versus patient Y that lives in the city, for example. They're going to be exposed to different levels of pollution, different air qualities, your living environment, what your house is like, dampness, all that sort of thing is going to affect it's going to affect the quality of the sample that you get, and also the diversity of the sample. Different people on different treatments, male versus female, different ethnic groups have different microbiota. We know that from the gut. Dietary factors. So that there's absolutely loads of confounding variables, and I think that then gets back to the point I was making, that probably the first steps are to do these carefully controlled experiments in mice, where you can limit the effect of these confounding variables, so that you can really specifically study what the microbiome itself is doing independently of all these other influences. Once we have that information, then we can, move into patients more and try and study what the sources of heterogeneity might be.
Fascinating. And you've talked about all those kind of compounding factors there, and with viral infections being so impactful. What have we, if it's not too early to say, what have we seen from COVID the past couple of years? Obviously, that's been our major respiratory problem. And what do you think we may see as the data comes in?
So I think the interesting thing about COVID, obviously, was that it affected specific groups more severely. So, young adults and children were not, were at very low risk of getting very sick, but elderly individuals, people with chronic lung diseases, people with other risk factors, were far more susceptible to severe disease. I don't think we fully understand why that is. It could be a sort of thing to do with, for example, the aging immune system. As we get older, our immune system works less well. That's one possibility. But the microbiome could be a major determinant. We know that the microbiome of an older person is different to that of a younger person. We know that the microbiome of patients with chronic lung disease, as I've already mentioned, COPD, is different to healthy individuals. So there could be a number of factors here at play that might explain why COVID-19 affected certain groups more severely. And I think microbiome is definitely one area. And again, it's something that through understanding how the microbiome affects responses to viruses generally, which is what we're studying, we would hope that in the future, if there's a SARS-CoV-3 or some other pandemic, that we might be better placed to be able to limit severity by hopefully targeting the airway microbiome.
Fascinating. And if we look to the future, but with a hopefully more positive lens than the next pandemic, if the gut microbiome, which we've drawn a lot of similarities from, can be improved with anything from probiotics and fermented foods all the way through to fecal transplants, what are the everyday and futuristic tips to supporting a healthy respiratory microbiome?
Yeah, so I think the ultimate goal of our research is to get to a point where you could give a patient an inhaler, and where they inhale bacteria similar to sort of fecal microbiota transplant, or altering the gut microbiome, so inhale either live or killed bacteria, or inhale bacterial metabolites. We know that one major way that commensal bacteria affect the immune system is through production of metabolites. So it could actually be that administering certain metabolites could be the answer. You may not actually have to put the bacteria in. But yeah, that's the sort of the long-term futuristic aim, is that we get to a point where we can start trialing these types of approaches. But obviously we need to understand what the friendly bacteria are that chronic lung disease king. And then the other sort of approach is kind of what we call a sort of sputum transplant, so similar to a fecal transplant. Can you wash out someone who's healthy's lungs, and then instill that into someone who has disease, to sort of re-engineer their disordered microbiome, and in doing so beneficially impact upon their immune system. And again, we're all probably a good 5 to 10 years away from being able to do that, but that would be the sort of long-term end goal, I guess.
Yeah, I would probably go for the inhaler to start with. And that's all full of potential there. Are there any other hopes for the future in regards to this field? You're so new, such a cutting-edge field.
Yeah, I think that would be the sort of what I described would be the main sort of approach that I guess we're working towards. I guess obviously in order to do that in the future, we're probably going to have to sort of do trials of administering very low doses of bacteria into people to see what's safe. So you need a kind of safety type study, because obviously it's a brand new approach that's not been trialed before. So everything would have to be done in a very safe way. And the other thing I guess to highlight is that, as well as the lung microbiome being important in lung immunity and the lung immune system, there's also a clear body of evidence that tells us that the gut microbiome affects the lungs. So there's something called the gut-lung axis, whereby changes in the gut microbiome can affect the ability of the lungs to respond to infections, and that's well described in the gut microbiome field. So again, people are looking at probiotics and all these types of things you mentioned in the context of lung disease, and we know that the gut microbiome of people with chronic lung disease is also altered compared to healthy people. So that could be a more realistic approach that could happen sooner than than manipulating the airway microbiome.
That's fascinating. And from a very sci-fi perspective, I'm seeing a personalized inhaler that you were talking about air pollution earlier. Maybe you end up with a certain type of microbiome enhancer if you live in one area versus another.
Absolutely. Yeah, that could be really interesting. Yeah, absolutely. Yeah, because undoubtedly pollution is a big cause of lung disease and lung infections. So yeah, you're absolutely right. We may need to get that specific, as you say, depending on where you live. You get given different types of bacteria.
And it shows that it's all connected as well, especially with the gut, which is absolutely fascinating. So thank you so much for joining me today. I feel like I've learned so much. I will definitely be signing up for the inhaler version. And I can't wait to see where the respiratory microbiome takes us in the future. So thank you so much.